Clinical Core webinar. So thank you for joining us this afternoon. So we'll be talking today on measuring vascular contributions to cognitive impairment and dementia, or VCID. So our ADRC Clinical Core group is trying to put more of these webinars, these discussions out so we can have more continued discussions building upon what we do at the ADC directors meetings for our Clinical Core meetings. So we've had one on health equity, disparities. There's been one other models of care talking about VCID today. So again, we'll be putting these out intermittently, so watch for those. I do want to thank Grace and Donnelly for her great assistance in helping to organize the meeting today, and of course to thank our speakers. So we have a great lineup today. So we'll be starting with, let's see if my slides will move forward. So we'll be starting with Angela Jefferson, who'll be talking about assessing cardiovascular integrity from the heart to the brain. Angela is the PI of the new Vanderbilt Exploratory ADRC, one of the new P20 grants. And so again, what we're trying to do is to think about, as the ADC program, how can we think about systemic vascular risk? How does it contribute to cognitive decline? And so not just in the brain, but systemically as well. So Angela will cover that. Then Dr. Donna Wilcock from University of Kentucky ADRC will talk about the potential of fluid biomarkers for clinical assessment and trials in VCID. And again, she's part of the MarkVCID program as well, which I'll touch on in just a minute. And then Dr. Konstantinos Arfanakis, and I'm going to ask you to, how do you pronounce your last name, Konstantinos? I always mispronounce it.
Arfanakis. That was pretty close. It
was pretty close, Arfanakis. So Dr. Arfanakis is from the Rush University ADRC and from MarkVCID as well. And he'll be talking about neuroimaging biomarkers of VCID. And then Dr. Charlie DeCarli will talk about an overview of diverse VCID, white matter lesion, etiology of dementia in diverse populations. And as many of you know, Dr. DeCarli is the PI and director of the UC Davis ADRC and also part of the MarkVCID program. And he has a new U19 grant that he'll be talking about at the end. And then we'll have time for discussion afterwards to think about how can we push our center program to better measure some of these vascular contributions using biomarkers, clinical assessments, etc. So again, I think many of us here know about the NIA Alzheimer's Disease Centers program. So again, they're shown here with the new exploratory centers shown in the states in the blue here. So we don't have blue and red states here. We have teal and green, I think, so instead. So again, you can see the states where we have some and then some with exploratory centers. We're thrilled to have those centers as part of the program as well. And then here on the right, you can see the MarkVCID Consortium Center. So there's some nice overlap with some of our Alzheimer's Centers programs. And so being able to share ideas across that, as we did at the Clinical Core Directors meeting back in the spring of 2019 as well. So we want to talk a little bit and give an update on where things are as far as the MarkVCID program, what some of the emerging biomarkers are, and then open up to discussion at the end. So we'll go ahead and move to our first presenter, and I'll stop sharing and welcome Dr. Angela Jefferson from our new P20. So welcome, Angela.
Thank you so much. I really appreciate it. We're delighted to be a part of the expanded ADRC network with our new P20 grant at Vanderbilt, and I appreciate the opportunity to present to the group today on the topic of cardiovascular phenotyping from the heart to the brain. I just want to mention that I'm a professor of neurology and I'm formally trained as a clinical neuropsychologist. So I just want to state at the outset that I am not a cardiologist. I just work with a lot of cardiovascular tools. Oops, sorry, hang on one second. That is not the right slide. Let's try this again. There we go. Okay, so our research program has a couple of interrelated areas of focus. First, we're interested in the impact of cardiovascular aging on Alzheimer's disease and cerebral small vessel disease. This cartoon illustrates cardiovascular aging from the heart through the aorta up to the carotids and into the cerebral macrocirculation and microcirculation, all of which we quantify in rich detail in our Vanderbilt Memory and Aging Project cohort, which we locally refer to as VMAP. While I won't get into the weeds today about the various results from our study, I do want to mention we found that subtle age-related changes in cardiovascular function and arterial stiffness affect cerebral blood flow, especially in the temporal lobes, as well as cognitive trajectory and risk for clinical dementia. We also have some recent and pending publications linking these cardiovascular variables to biomarkers of core Alzheimer's disease pathology, as well as what I call concomitant pathological pathways, such as neuroinflammation and synaptic dysfunction. And these observations, importantly, are in the absence of prevalent cardiovascular disease. A second area of focus is delineating the etiology and clinical consequences of cerebral small vessel disease. In VMAP, we're quantifying supravascular structure and function in rich detail, so we can ask questions like, do certain small vessel disease markers have a greater impact on cognition than others? Today, I'm going to focus on this first area of emphasis in cardiovascular aging. I'll highlight some disparities that we see within VMAP regarding participants' self-report and their actual risk factor status. I'll walk you through the vascular tree and highlight a few easy to implement assessments that we work with in VMAP. And I'll review some multivariable vascular risk factor scores that are easy to add to any clinical research project. Why should we measure cardiovascular health in detail when it's so much easier and free to rely on self-report? Here are some data from VMAP. This is a growing cohort of adults aged 50 and older living in Middle Tennessee, Southern Kentucky, and Northern Alabama. At enrollment, 284 of these participants self-reported that they did not have diabetes. However, at the same visit, when we looked at their hemoglobin A1C levels, we found 2% of those individuals had levels in the diabetic range, and 20% had levels in the pre-diabetic range. Basically, 1 in 5 had diabetes or pre-diabetes and didn't know it. And that's not really an overwhelming number. But let's look at the cholesterol data. 152 of our participants reported they had no history of cholesterol problems. However, we found from their blood work at study enrollment that 57% of those individuals had LDL values above 110. What happens when we look at something easy to capture like systolic blood pressure? 162 of our participants reported being normotensive. They told our team they had no history of problems with elevated blood pressure at enrollment. However, 23% of them had high normal systolic blood pressure values between 120 and 130. 25% had systolic blood pressure values that would be considered pre-hypertensive between 130 and 140. And 41% had outright hypertension, that is systolic blood pressure values over 140. So out of everyone who told us that their blood pressure was normal, nearly 5 out of 10 were high normal or pre-hypertensive. 4 out of 10 were outright hypertensive, and only 1 out of 10 was correct about being normotensive. The take-home message here is that self-report grossly under-identifies the presence of important vascular risk factors, some more so than others, and certainly preclinical or subclinical risk factors. And
is that self-report informed by a clinical exam by a cardiologist?
No, this is basically filling out a medical history form that is reviewed by our nurse.
So it's an uninformed self-report.
Right, but they haven't undergone a detailed assessment. They're just being sort of screened in the community.
Sure. I just want a little
no, that's a great question.
Little granularity.
Yep, that's a great question. I do also want to briefly just address the generalizability of the information to other cohorts in the general population. Our participants have a mean education level of 12 years with a median post-high school reading level. I suspect that these numbers would be even more striking in a cohort with lower education levels, less health literacy, or barriers to healthcare. So what should we be measuring along the vascular?
Angela,
yes.
Charlie here. So we've created our own scale, and one of the things we discovered is that it has to do with how you ask it. So if you give a whole series of terms, one thing we've discovered is that what you would call hypertension, that's not true about the H1C, we haven't looked at that, but questions like hypertension, it's incredible what people get told, and the more options you give them, the better. And
the more you capture.
Yeah, we've confirmed this with the MR data. So I think that's one of the tricks.
I also think that, you know, when you're calling something diabetes versus sugar.
Yeah, exactly.
Right? Yeah, that absolutely. I think that that's absolutely true. And like I said, like those data are just representative of a high school educated, you know, post high school reading level, reasonably sort of, you know, affluent group of individuals. And so I think it'd be highly variable across other populations as well. It's like subjective cognitive decline. It depends on how you ask it. So what should we be measuring within the vascular tree? Well, we can measure a lot of different things. And going back to this illustration of cardiovascular aging, here's just a subset of what we measure in VMAP. So we capture clinical blood work, blood pressure, cardiac structure and function, aortic compliance, neck flow velocity through the carotids, macro vessel wall imaging to quantify lumen diameter and wall thickness, patency of the circle of Willis, cerebral blood flow and reactivity, and oxygen extraction fraction reflecting the amount of oxygen in the venous return. And so this level of phenotyping is beyond the scope of a common or easy to implement protocol, but it highlights that we have the capacity to connect a variety of systemic vascular health markers with detailed cerebrovascular variables to better understand the interface of cardiovascular aging on the brain. Subsequent presenters are going to cover aspects of neuroimaging, so I'm going to focus my time on these systemic elements. So first, clinical blood work offers important data on cardiovascular risk factors such as cholesterol, A1C, glucose, and high sensitivity C-reactive protein. Our blood draw is fascinating because we also collect insulin. And to capture all of these markers listed here for our CTSA Clinical Research Center space fees, we pay about $200 per participant. At my prior institution, we used Quest Diagnostics, and it was much less expensive. So there is some variability in terms of capturing blood work across institutions. Every participant in our center has an echo, and this is our sonographer, Joanne Gottlieb, who's employed by our institution's CTSA Clinical Research Center. From each echo, we collect cardiac structure and function, including left ventricular mass, ejection fraction, and many other variables. And this is one of our least expensive protocols to acquire. It only costs $35 per echo because the rate is subsidized by our CTSA. We also pay a board-certified cardiologist to overread Joanne's measurements, but that cost is nominal as he's shared across multiple projects. And we use a percent effort model given our heavily integrated program with cardiovascular medicine, but it can also be easily integrated into clinical workflow at most institutions. And then every participant in our study also gets a cardiac MRI, which provides some of the same information as the echo, but at a much higher resolution. Plus, it provides important information about aortic stiffness. So using cardiac MR, we can capture age-related changes centrally at the source of the aorta, rather than making inferences about it from peripheral vascular beds. The problem is this is one of our most expensive protocols. Acquisition costs more than $800 per participant, plus we finance all the processing costs performed by our radiology informatics colleagues. There are cheaper ways to capture stiffness, such as tonometry and Doppler, but for reasons I won't go into today, we prefer the precision of measuring central stiffness in the aorta. We also focus, I think, pretty often on what I would call silo analyses, where we consider one vascular risk factor variable at a time, such as hypertension, in isolation from other frequent comorbidities. However, there's potentially a lot of utility in integrating multiple pieces of information to capture shared or unique effects of vascular risk factors on brain health. So I want to run through a few options. So I trained and worked at Boston University for a number of years before coming to Vanderbilt. So I've used the Framingham-Stokes profile over the years, and this tool offers a 10-year probability of incident stroke with weighted points assigned by sex for a series of risk factor variables. And we've used this as a predictor of cognitive trajectory within the NAC data set, but we've also used this as a vascular bundle covariate in many of our analyses. Another tool I want to highlight is the ASCVD risk estimator. It's probably among the most commonly used tools and offers 10-year risk information for atherosclerotic cardiovascular disease. But depending on the demographics and general health of the particular cohort you're interested in studying, different risk factor scores may be more or less accurate in quantifying risk. For example, the Framingham Coronary Heart Disease score offers 10-year risk of myocardial infarction or death, and it has the added advantage of being validated in individuals as young as 30 years of age, which is important as we think about how midlife exposures have a major impact on late life cognitive health. Another example, the Reynolds risk score was originally validated in women, so its predictive value is a bit better in females compared to males. The Q-RISK-3 is very comprehensive and considers additional systemic comorbidities that are really important, like kidney disease or autoimmune disease. So while it's the most complicated to calculate, if you have a cohort that has a lot of comorbidities, it may be a much more accurate estimate. And then the heart score is probably the simplest to calculate and based predominantly on objective data rather than self-report. It could be a good place to start if you want to capture the shared effects of vascular health, but you can't implement echoes or other protocols. So bringing things back to the vascular tree, I reviewed earlier that we can capture a number of these different things. What we can sort of easily capture, I think, is really blood pressure, clinical blood work, and maybe even cardiac structure and function using echo for a modest cost. These assessments yield variables that we can use individually or in combination to formulate multi-risk or multivariable risk scores to better integrate cardiovascular aging issues into our Alzheimer's disease research activities. And I just want to acknowledge the funding sources and colleagues, especially our cardiology and radiology informatics collaborators who helped us implement many of these protocols. And I hope you all can visit us in Nashville when social distancing is a thing of the past. In the meantime, thanks so much for your attention.
Thank you, Dr. Jefferson. That was wonderful. So we are going to hold questions till the end of our presentation, but thinking, integrating what we're hearing from Dr. Jefferson with some of the other thoughts we'll be talking about. So our next A speaker is Dr. Donna Wilcock from University of Kentucky, Sanders Brown Center on Aging, the Alzheimer's Center, and MarkVCID program. So she'll talk about the potential of fluid biomarkers for clinical assessment and trials in VCID. So welcome, Dr. Wilcock.
Yeah, hi, good afternoon, everyone. For most people anyway, it's after 12 on the West Coast. So yeah, good afternoon. It's great to be with you all. I want to talk about some of the fluid biomarkers that we're looking at in MarkVCID. And then towards the end, I'm going to talk about one of those biomarkers in a little bit more detail, what biological meaning it may have. So I'm going to jump straight into the kits. I think most people are familiar with MarkVCID, so just briefly, MarkVCID is a NINDS and NIA-funded consortium that composes of 7 sites around the US. The overall goal of the consortium is to both discover, identify, and cross-site validate biomarkers of small vessel disease, related vascular cognitive impairment. I think Konstantinos is going to speak to you about the imaging biomarkers, at least some of them that are being validated in MarkVCID. I want to focus today on the fluid biomarkers that MarkVCID is looking at. We're in our 4th year now, so prospective recruitment started probably about 18 to 24 months ago for most sites, and I think the majority of sites now have reached full recruitment, and we're coming up on first annual visits. So we're starting to reap some fruit from this wonderful consortium. We're overseen by a coordinating center, which is led by Steve Greenberg at MGH. So the first fluid kit is called the Endothelial Signaling Kit. And this is a plasma-based kit that composes 3 plasma proteins, VEGF-D, which is not the prototypical VEGF. That would be VEGF-A, the one when we really think about what VEGF is. So VEGF-D, PLGF, this is placental growth factor, and I'll come to that a little bit more later, and BFGF. These are 3 of, I believe it's 7 proteins that can be assessed on the mesoscale discovery VPLEX angiogenesis kit. The mesoscale discovery is a highly sensitive electrochemiluminescent ELISA that is capable of multiplexing samples. The rationale for the kit is that endothelial dysfunction early in cerebrovascular disease causes compensatory upregulation of endothelial and angiogenesis signaling. Preliminary data for this kit primarily came from Joel Kramer's group and Charlie DeCarli's group, looking at longitudinal data showed that baseline signal in the 3 markers there predicted accelerated white matter injury and cognitive decline. The cognitive decline was measured by a composite memory score. Additional cross-sectional data showed that the kit, the 3 proteins, when you take the z-scores of those proteins, is associated with higher cerebral free water and lower whole brain fractional anisotropy on neuroimaging. And this was even after you control for the presence of amyloid on PET. This is currently still going through a step that we call instrument validation. All of the kits go through instrument validation before they go through the biological validation. And what this is ensuring that the sites that are participating in the validation are able to measure the same levels of these proteins on their instruments in the same samples. And so we're just going through some calibration steps before we can complete that instrument validation. And Jason Hinman at UCLA is the lead on this kit. The next kit that is being validated it's termed the Endothelial Inflammation Kit. This is using plasma exosomes and is led by Fanny Elahi at UCSF. So this is a composite biomarker kit that measures 3 key complement proteins in endothelial-derived exosomes. And the idea is that endothelial inflammation is likely an early stage of cerebrovascular disease. The preliminary data showed that you had significantly higher levels of the complement proteins in exosomes from individuals with white matter hyperintensity signal on MRI as opposed to those that did not have a WMH signal. And so you can imagine isolation of exosomes from plasma is a very labor-intensive step. And so the validation that's occurring right now is really trying to get sites to be able to perform this exosome isolation with a good consistency. And Dr. Elahi is working very diligently to try and get this up and running, because I think it's a very exciting kit. The next kit is what we're calling the CSF Stratification Kit. And really this is a CSF PLGF kit. Everything else, as you know, is pretty well validated in AD and dementia. But what we don't have in CSF is a small vessel marker. So PLGF, or placental growth factor, we've seen really good relationships between PLGF in the CSF and in the plasma and white matter hyperintensity. And this was replicated in the UK cohort here and in the UCSF cohort with Joel Kramer and Fanny Elahi. So the requirement, the premise of this is that if you're going to have a trial either for small vessel disease or Alzheimer's disease, we not only need to confirm that the targeted pathology is present, but we also need to minimize those comorbidities wherever we can. So the goal is to use this PLGF as an additional CSF biomarker to stratify patients. Instrument validation on this kit is complete. 3 sites have run the same samples, and we got a very nice interclass correlation coefficient of 0.94. Our threshold was going to be anything over 0.8, so that was great. This data here shows baseline white matter hyperintensity volume and the association of PLGF in the CSF with that, and I'm the lead on this kit. Finally, neurofilament light in the plasma is being measured, and this is led by Dr. Seshadri out of UT Health San Antonio. And what she's shown in the charge cohort is that higher plasma NFL concentrations are associated with increasing white matter hyperintensity burden and poorer cognition using several different cognitive outcomes. NFL remained predictive of cognitive performance in individuals with low white matter burden, which you would expect given the large body of NFL data out there right now. And cross-site instrument validation has been complete on this, and they achieved an interclass correlation coefficient of 0.91 across 3 sites. I should mention both the NFL and the prior PLGF kit is using the Quanterix Simoa platform. So I just want to talk a little bit about PLGF. We got very interested in this. You know, it's not really something you would think about when you think about dementia. It's a member of the VEGF family, and when it's on its own, it's pretty weakly angiogenic, but when it's part of a heterodimer with VEGF-A, it is particularly potent at stimulating angiogenesis. PLGF deletion in mice does impair angiogenesis in response to some pathophysiological conditions. So it doesn't seem to be involved in normal angiogenic responses, and these mice can breed and they develop just fine. But if you induce ischemia, and nobody's really looked at the brains of these mice, but like muscle ischemia or heart, they have a poor angiogenic response. This is just showing you our data where we see an association both with CSF and plasma PLGF and white matter hyperintensity. So we wanted to look at where PLGF might be expressed in the brain. And this is some immunohistochemistry showing PLGF staining in the white matter of, white matter of age-matched control versus an individual who died with severe small vessel disease. You see that there's a lot of perivascular labeling. Also, throughout the parenchyma, there appears to be some glial labeling. We believe this to be astroglia, but we're doing double label to confirm. And that perivascular staining may actually be parasites, but again, something that we're following up on. We found in a set of matched autopsy tissue, this is 12 per group, that individuals with small vessel disease had higher PLGF in the plasma. They had higher gene expression of both PLGF and VEGF-D compared to age-matched controls, and the brain protein level of PLGF was significantly higher. And then just to finish up, Dr. Elahi and I have been doing some collaboration. We were interested to see what cells are driving the PLGF increase, and it looks to be astrocytes and endothelial derived. This is exosomes isolated from the plasma, but neuronally derived exosomes did not seem to express, did not seem to have any PLGF associated with them. So in summary, fluids do seem to provide us an opportunity to identify biomarker profiles. And unlike our PET capabilities, fluid biomarkers can allow us to identify the presence of many pathologies. 3 of the 4 kits that you just saw that have advanced to cross-site validation in MarkVCID are focused on endothelial responses. And I really think this may afford us an opportunity to look for novel therapeutic targets. Endothelial biology seems to be key in small vessel disease. And fluid biomarkers are probably going to be our optimal method for tracking target engagement and efficacy in trials. And I think this is where we're going to see the utility over time. So with that, I'd just like to thank the folks in my lab and the folks at the ADRC here, our outside collaborators. And I just want to say the whole MarkVCID team, because I just can't list all of you on here, but we're working great together, and it's been a fun ride so far. So thanks for your attention.
Thank you, Dr. Wilcock. That was great. And it's such amazing to see the progress that's been made even since the time we presented back in the spring of last year. So thank you for giving us that update. And again, keep your questions, think about your questions, go ahead and put them in the chat if you'd like, or we will have time at the end of all 4 presentations to have a good discussion. So we're thrilled to introduce our next speaker is Dr. Konstantinos Arfanakis from Rush University ADRC, and he'll be speaking on neuroimaging of VCID.
Thank you for the introduction. All right. So in today's presentation on the neuroimaging biomarkers of VCID, I'm going to be focusing on the ARTS biomarker, the biomarker that we've developed for predicting arteriolosclerosis pathology. Arteriolosclerosis, brain arteriolosclerosis is one of the main pathologies of cerebral small vessel disease, and it involves thickening of the vessel wall and stenosis of arterioles. Arteriolosclerosis is common in older persons, is more severe in women and in Black older adults, and has been associated with lower level of cognitive and motor function, higher rate of cognitive decline, greater sleep fragmentation, higher odds of Alzheimer's dementia. In spite,
we might need to have you switch. I think it's still on your intro slide, so you might have to switch which screen is being shown, so it'll follow along with where you are.
So you're seeing the
in the introduction slide still.
Oh, you're seeing the introduction. Okay, let's see why that's the case.
If you have to switch screens, possibly.
You don't see the. So you see the title slide. You're saying.
Yes. Mm-hmm.
Did it switch now or no? Still the same?
No, I think you may have to back out and then come back in again.
Okay, let me try
just
that.
Make sure it's on the right. If you have two screens, it might be on the other screen. That was, yeah, that took us to the, let's see. And I think if you switch screens, that'll.
Let me see. Okay, so did it go to the next slide or no?
No.
No. Okay, let me try the swap displays. What do you see now?
Still the first introductory screen. If you go to the top and click of your PowerPoint and say which show, are you in presentation mode?
It says sharing is paused. Bring your shared window to the front.
Maybe if you could just go, why don't you just go ahead and back out of presentation mode and just, there it is, but now you have to switch screens where it says swap displays at the upper
Okay,
left.
yeah, I'll do that.
And then,
I don't know why it, okay, let's see, where's my mouse? Sorry about that.
oh, you're fine. There we go, perfect, thank you.
All right, okay. So as I was saying, arteriolosclerosis is a thickening of the vessel wall and stenosis of arterioles. It's common in older persons, more severe in women and Black older adults. It has been associated with a lower level of cognitive and motor function, higher rate of cognitive decline, greater sleep fragmentation, higher odds of Alzheimer's dementia. In spite of its prevalence and detrimental effects, arteriolosclerosis can only be diagnosed at autopsy. So the goal of our interdisciplinary team at Rush University and Illinois Institute of Technology has been to develop an MRI-based biomarker of arteriolosclerosis. To do this, it has involved a number of steps. In the first step, which we call training, we've trained an ex vivo classifier of arteriolosclerosis in the Memory and Aging Project and the Religious Order Study. We then translated the ex vivo classifier of arteriolosclerosis to in vivo and developed the in vivo biomarker, which we call ARTS. We then tested the performance of ARTS in predicting arteriolosclerosis pathology. We also tested the hypothesis that the higher ARTS score is associated with faster cognitive decline 2 years after MRI. And then we took this hypothesis and we tested it also in a different study in Mars, in the Minority Aging Research Study. And this serves as the validation step 1. And then we also tested the same hypothesis in ADNI 2 and 3 participants. And finally, in MarkVCID, we've been working currently on the testing intra-rater, inter-rater, and scan-rescan reproducibility. So let's see the steps one by one. In terms of the training, we've used ex vivo MRI and pathology data from 119 MAPROS participants. On average, their age was 91.1 years at death. 33% were NCI, 25% were MCI, and 42% were demented. We then trained the logistic regression classifiers involving a set of features that are listed here: white matter hyperintensity features, DTI features, and also demographic features. And we tried different combinations, and we saw that by combining all 3 sets of features, white matter hyperintensities, DTIs, and demographics, we get an AUC, we get a performance of 0.78%. which is represented by this black curve here. When we combined only DTI with demographics or only wide matter hyperintensities with the demographics, we got a lower performance of 0.73%, and when we used only demographics, we got an even smaller, even lower performance. So we decided to develop this ex vivo classifier using all three sets of features. We then translated this ex vivo classifier to in vivo by combining in vivo and ex vivo MRI data on the same MAPROS participants. And we developed the software that runs the ARTS biomarker in a way that makes the whole thing fully automated. Currently, the ARTS biomarker takes as input DICOM or nifty data, so data straight out of the scanner. It requires three types of images, 3D-T1 weighted images, which can be collected either with MP-RAGE or multi-echo MP-RAGE or with FSPGR. It requires FLAIR, which could be 2D FLAIR or 3D FLAIR, and requires also DTI, which could have reverse polarity data along with it or not. The output of ARTS is a score that represents the likelihood of arteriolosclerosis. The software is a singularity container. Essentially, you just need to download it and you don't need to install any other software to support running ARTS. So you don't need to install FSLs and SPMs and MATLABs, etc., etc. So you just download the ARTS container and it runs, and it runs on Linux, on Mac, probably on Windows. We haven't tested that yet. And it also works on personal computers or high performance clusters. So we then tested the ARTS biomarker, the performance of the ARTS biomarker in predicting pathology. For that, we used in vivo MRI and pathology data from 79 MAPROS participants. None of these participants were part of the training group, which is shown here in green. So these were MAPROS participants, but a different set of MAPROS participants, and it was in vivo MRI data. This group was about 2 years younger than the training group, and it was mainly NCI or MCI. Only 9% were demented, and if we compare this to the training group had 42% of dementia cases. So let's see what we got. We got something quite interesting, I believe. So when we only test using participants that have short ante-mortem intervals, so the interval between in vivo MRI and death is less than 2 years, 2 years or less, the AUC that we got is 0.79 or 0.8, which is almost identical to what we got ex vivo, which was 7.78. But as we started considering participants with longer ante-mortem intervals, the performance dropped. And we believe the reason for that is that ARTS is looking at a relatively healthy version of the brain. Then 6 years go by, and autopsy is looking at the more diseased part of the brain. So we're kind of ignoring what happens down here, and we're focusing on this part here, which is giving an AUC that's very similar to that obtained ex vivo. We also plotted the ARTS score as a function of arteriolosclerosis severity diagnosed pathologically, and we see that the ARTS score increases nicely with when you go from no arteriolosclerosis to mild to moderate and severe. We then tested the hypothesis that the higher ARTS score in non-demented older adults is associated with a greater decline in cognition 2 years after MRI. And we did this testing in 3 groups, in MAPROS participants, in MARS participants, and ADNI 2 and 3 participants. These groups are about 10 to 14 years younger than the training group, and they have no demented participants. The MAPROS and MARS are almost exclusively NCI, almost. The ADNI 2 and 3 participants are more evenly split between NCI and MCI. And also should point out that MARS has exclusively Black older adults. So what we saw in MAPROS was that a higher ARTS score in MAPROS was associated with a greater decline in global cognition and perceptual speed. Also semantic memory and working memory, and episodic memory and visuospatial abilities, but the strongest association was with global cognition change and the perceptual speed change. In Mars, we saw association of a higher ART score is associated with a greater decline in perceptual speed. And then moving on to ADNI, we saw that a higher ART score, and I'm looking at a different screen because there is some other window overlapping on the right. In ADNI, we saw that the higher ART score is associated with a higher change in ADAS-COG, trails B, trails A, and the ADNI executive function composite score and ADNI memory function executive score, composite score. Now, because we have multiple comparisons here, we performed the Bonferroni correction. And what we saw, that what survived was this association of ART score with decline in global cognition and perceptual speed in MAPROS, and in ADNI with increased trails B score, which is very encouraging because perceptual speed and trails B essentially cover very similar cognitive abilities. And these are related to SVD. The association did not survive, the perceptual speed association did not survive in Mars, it did not survive the Bonferroni correction, but we're not really worried about that because the Mars sample was relatively small. The good thing is that before the Bonferroni correction, perceptual speed was what was showing up. So we're testing the same hypothesis currently. There's a lot of ongoing work on that, testing the same hypothesis in MarkVCID. We also looked at the inter-rater reproducibility of ARTS. So 7 MarkVCID sites run ARTS on the same 20 data sets. And these are the scores from the different sites. You see they're almost identical. The ICC is ridiculously high, 0.99997. And when we look at correlations of ARTS scores across sites, the correlation is also very high. We also looked at the scan-rescan reproducibility of ARTS at 7 sites. Here, 7 sites imaged 5 or 6 participants twice within a week. And what you see here are the ARTS scores from visit 1 and visit 2. Again, you see some great agreement between the different visits. And again, the ICC was very high at 0.998. So the take-home messages are that brain arteriolosclerosis is common in older adults and it's detrimental. Arteriolosclerosis, unfortunately, currently can only be diagnosed at autopsy. Therefore, our goal has been to develop a biomarker, an MRI-based biomarker of arteriolosclerosis, and we've developed software that makes it fully automated. The big advantage I feel of this biomarker is that it was trained on ex vivo MRI and pathology data, which has a major advantage of eliminating essentially the time interval between imaging and autopsy. The alternative of using in vivo MRI and pathology requires very short antemortem intervals. If the antemortem intervals become slightly longer, then we have additional pathology developing, and at that point, MRI and autopsy are looking at 2 different brains, and that can affect negatively the development of biomarkers and the testing of biomarkers. In our case, it may have affected the testing that we did. Also, ex vivo MRI and pathology suggests that imaging is done independent of frailty level. Everybody gets imaged, so it may enhance generalization. The performance of ARTS in predicting arteriolosclerosis pathology was 0.78, both ex vivo and in vivo. And we showed an interesting association of ARTS score with 2-year decline in perceptual speed in MAPROS and with increased trail score in ADNI 2 and 3, which is again encouraging because they are looking at similar cognitive abilities. And in terms of inter-rater and scan-rescan reproducibility, both of them were particularly high. The high inter-rater reproducibility means that if we all process the same data, we will get the same score. The scan-rescan reproducibility means that ARTS is relatively stable, is quite stable, which means that it's very appropriate for longitudinal studies. And finally, I would like to mention that ARTS will be made freely available in the next month or two. And with that, I would like to thank the participants of MAP, ROS, MARS, and ADNI. I would like to thank our funding sources and also the multidisciplinary team at Rush University and Illinois Institute of Technology, including neuropathologists, neurologists, biomedical engineers, neuropsychologists. And also I would like to thank you for your attention.
Thank you so much, Dr. Arfanakis. That was a great presentation. We will move on and save our questions to the end, but please do use the chat function to enter your questions. I think that'll help us get through a little more questions at the end, because we'll be a little short on time for questions at the end. So please enter in the chat function. Our last presenter is Dr. Charlie DeCarli. He'll be speaking on overview of diverse VCID, white matter lesion, etiology of dementia in diverse populations. So Dr. DeCarli. You're on mute, Charlie. Let's see if I can.
Sorry about that. It's really a pleasure to have the opportunity to talk to you all about a new program that we're developing. There's no data yet. We're just putting things together, but I thought I'd give an overview since I will be asking many of you to participate in this project. This is a U19 in collaboration with NINDS primarily, but with NIA as a secondary sponsor. And my co-PI is Miriam Fernage, who will be doing the genetics part. The goal of what we're trying to do here is pretty simple. I think the clinicians in the room probably know this very well, and that is that you often see people with minor cognitive complaints. They say they're having trouble with their memory. You see some smudge on their MRI and you say to yourself, how important is that? And this will be the first test that will look at the impact of these baseline and progressive differences in white matter injury, including of the entire spectrum on cognition, and then taking this into a precision medicine approach, much like what Angela was talking about, that is to form a kind of a white matter risk or a VCID risk scale. The specific aims of the grant, I'll go over quickly, is to identify the extent and characteristics of white matter disease that influence cognitive and health outcomes. I point out that we include health outcomes because we want to use hard outcomes as well as cognitive outcomes. And these are three hard outcomes, which are stroke, MI, and death. We'll look at that by using various MRI and vascular risk factor markers, polygenic risk scores, and also novel blood biomarkers that Donna talked a little bit about. The second is to look at the progression and how they have of these white matter injury factors on health outcomes. Same thing, just in a longitudinal design. And then finally, based on the first two aims, we want to build and validate a predictive risk score model. And this would help to say, okay, if in the setting of you walk in the office with cognitive complaints, you have some white matter smudge, how much does this matter to your outcome over the next 3 to 5 years? And a second part is that we will validate this precision medicine approach using a host of epidemiological studies with that Miriam and I are participants. So the concept here is that we have causal factors, progressive white matter injury, and various outcomes. The causal factors are the common things that you would think of: age, vascular risk, concurrent vascular disease, AD pathology, and genetics. And then we'll use a spectrum of analytical tools to look at baseline and progressive vascular injuries or white matter injury that goes beyond just looking at white matter, but the spectrum of white matter injury, as well as cerebral microbleeds, incident MR infarcts, leading to a change in cognition or a change in diagnosis, such as from normal to mildly impaired to dementia, or mildly impaired to dementia. And then these three hard outcomes of stroke, MRI, and death. We're fully aware, though, that there are moderators and mediators that are likely to interact on this pathway, including race, ethnicity. A unique aspect of using Life Simple 7 to look at control of vascular risk, sex, AD genetics, AD biomarkers, inflammation, and endothelial dysfunction, which is very consistent with what Donna was speaking to with her presentation. The timeline and milestones, we just got funding about a month ago. So we're into the development where we will be developing IRB protocols or SOPs, site selection and training. It will go then for 5 years. So this is a 6-year project. And it's hope that we'll have baseline and 2 follow-up examinations over this time period. We plan to enroll 2,250 individuals. The inclusion criteria are really quite straightforward. You have to have a cognitive complaint, and I'll show you that in a second. Incidental white matter hyperintensities, and then we are stratifying to be diverse under Black, Hispanic, and white. Exclusion would be clinical stroke, the presence of large cortical MRI infarcts, or dementia at baseline. The assessments will involve a standard ADRC assessment with some subtle enhanced cognition, the Framingham cardiovascular risk, the vascular health questionnaire that we developed, Life Simple 7, clinical laboratories, blood, plasma, DNA, MRI, and then those who may die along the over the course, we may have some autopsy results. The goals are the clinical progression, a change in diagnostic, white matter progression, and the hard outcomes. And the deliverable of this study will be this clinical risk score. What do we mean by cognitive complaint? It's very simple. And unfortunately, this is going to be ubiquitous, but it's the best we've got right now. And I think that it was alluded to earlier about, subjective cognitive complaint can be quite variable, but we settled on this because of its validity. Have you experienced a decline in your memory or other aspect of thinking in the last 1 to 3 years? And this conforms to the International Subjective Cognitive Decline Initiative work groups and has been used by a number of large studies. This is a very busy slide, but this is just to say that we have 4 cores that will be working on this. Most importantly for this group is that our recruitment and retention core is designed to support the AD Centers program, as well as the MarkVCID consortium, which now virtually is overlapping. We will be looking for MRI, blood, and clinical that will go to Lonnie, to our coordinating center, and to NAC, where we will do a storage and processing, develop summary measures. These will go into a data repository, and there will be a resource sharing portal, which we will then give back these summary measures, AD and white matter genetic risk scores, along with the ATN biomarkers. So for you all, what we ask and will pay for is we'll pay for recruitment and clinical evaluation as part of your clinical core. We've already identified 10 sites with diversity experience. We will pay for an MRI under the SCAN protocol, blood, UDS neuropsych, vascular questionnaire, list learning tests will be the additional, along with Life Simple 7. But just to show that there will be a couple of just minor additions to a standard clinical core evaluation. What will we give back to you? quantitative MRI summary data, which will include white matter hyperintensity, hippocampus, ventricles, regional gray matter volumes, regional cortical thickness, much like you would see with a free surfer kind of analysis. The ATN markers, which will include A beta 4042. Right now, and these will be on the Quanterix platform. So right now, Quanterix has 181, but they are looking into adopting the 217. And these genetic risk scores, both of CVD and AD. Also, the way this will work will be and emphasize that there will be a resource sharing protocol that will be given. All this data will be given back to NAC and to the sites. LONI will serve as a resource sharing portal as well as our own. But the goal here, and I want to finish on this last, is that this is to support future collaborations in ancillary studies. And then, of course, we will do the primary analysis, which will be this risk scale. So we're looking also to encourage you all to give us ideas about ancillary studies and to promote this. We wrote as part of this U19 is that there will be a funded pilot project program of $100,000 a year for years 3 through 6 for innovative projects and the data sharing portal. So I kept this brief just so that to give you a kind of a quick overview of what we're proposing. More will be coming, and you will be seeing me at some of the ADRC meetings presenting this in more detail. And I appreciate your attention. Thank you.
Thank you so much, Dr. DeCarli. I know some of you put questions in the chat and Dr. Arfanakis is answering those, but we do have a few minutes for questions.
Can I ask a question live?
Yes.
And
Sure.
that's just very, very good, Charlie. I wonder, you say NAC and LONI are sharing the data. Are they sharing different data or the same data?
No, it'll all be the same data, John. Thank you for pointing that out.
Okay, okay.
They will be the portals which will receive the data. NAC will be the opportunity to get the summary data, and LONI will be a place where you can, like Scan, will get the raw data if you want it.
And the next question is, how did you get 6 years? When we got our U19, Austin Yang said I'll be able to get mine renewed for another 30 years. I didn't tell him that I was 72 already, but how did 6 years is unusual for a grant? Was that something specific to the study?
Yes, this was written into the grant because of an experience with the MarkVCID.
Okay,
where
good.
The first year is used as an organizational.
Yeah, it's unusual. Maybe you'll get it for another 30 years too.
Right, we have time for maybe just 1 or 2 more questions. So any other questions? We do have some answers in the chat, and also please note that Grayson has put the presenters' email addresses in the chat. Sorry, presenters, if you didn't plan on that, but no. But that way you can directly talk with the presenters as well. But other questions people have, if you want to unmute and share.
I have a question for Charlie, just about, you mentioned the 10 sites that have been identified. Is it restricted to 10 sites, or will you be looking for more sites?
No, we will be talking about opening that up. And so, yes, there are sites that have certain capabilities that will be prioritized, but we are looking to open it up to the entire ADRC cohort.
Great, other questions? Maybe Angela will ask, what do you think of the marker of the questions that look at composite vascular risk, the ones that you showed? What do you think are best suited for the ADRCs and most representative across diverse patient panels, patient cohorts?
Well, I think, there's just a lot of different options out there, and I feel like some of the risk scores are similar to serial list learning measurements, where everyone has their favorite, and they have their favorite for a different reason. Charlie is using one, we use one. That's a little bit of a problem. I think, from my perspective, I think one of the most important things is trying to quantify really clearly the variables or information that are inputted in. Like that's the more important thing, is making sure that you're capturing the variables correctly and as accurately, precisely as possible. Charlie, I know you have opinions on this as well, but I think that's the sort of more important piece, like just kind of, because some of them are just, are like some of them having the younger age groups, like could be more important if you're looking at people in their 50s, whereas like the FSRP is for, I think it starts at 54 or 56. And so they're different sort of nuances to them. I think the accuracy of the input variables is the most important component.
And Cindy, I think one other thing to keep in mind is that we tend to conflate risk and consequences. So I like to think about, you know, risk factors as something that is not a disease, and then the consequences, which are diseases. So you can have hypertension, but not have chronic renal failure, for example, or have an MI. And so in my mind, that's a different person. And so I like to keep those things in mind. But one, I think that the Clinical Core Task Force could do is thinking about looking at these and coming up with a good way for us to use these best to come up with a unified scale. And I know that Angela would be helpful in that regard. And I, of course, I'm always one to have an opinion.
There is a new, as far as accuracy goes, there is a new blood pressure module that NI's developed for the UDS. So the blood pressure will get measured in a standardized way across all the centers, which is great. So that's a great addition. So that reflects what you were talking about.
Yep. Yep.
I think we're at the top of the hour, so we're going to have to stop here. But I just want to thank so much all four of our speakers for presenting today. Again, I think this isn't stopping the conversation now. It's just going to continue. So hopefully continue via email, discussions at meetings. And again, watch for other Clinical Core webinars that'll be coming through the internet, sponsored by NAC and NIA. So thank you all so much for being here today.
Nice job, Cindy. Thank you.
Thanks, Cindy.
Bye-bye.